My name is Maryan Zirkle. I'm the Executive Director of the BRAINCommons at Cohen Veterans Bioscience, or CVB. CVB is a nonprofit 501c3 biomedical research and technology organization. We're dedicated to advancing brain health by fast-tracking precision diagnostics and therapeutics. At CVB, I'm responsible for the oversight and development of our flagship enabling platform and supportive data sharing. It's my pleasure to be presenting to you on how the BRAINCommons is paving the way for pan-cohort analysis. Cohen Veterans Bioscience, or CVB, has the ultimate goal of prevention and personalized medicine for brain health. Our approach is to build enabling platforms with strategic partners and to adopt a team science approach to fast-track solutions in years, not decades. We are in a new era of data-driven biomedical informatics research, where organizations with powerful data acquisition technologies are generating health data at increasing scales. With all of this high-quality health data available, our biggest challenge is harnessing the combined power for accelerated discovery and impact. Harnessing this data requires a next-generation digital ecosystem that can scale to support the massive amounts of data and enable discovery across disparate cohorts. CVB's answer to this unmet need is the development of the BRAINCommons to democratize brain health data. The BRAINCommons is a one-stop shop for data integration and analytics for researchers and data scientists. We offer a secure infrastructure providing 3 levels of access to de-identified multimodal data. This data is curated and standardized to a brain disease common data model that is leveraging widely used models and standards. The BRAINCommons has a cloud-based architecture for big data capacity, and our data partners control their level of access for the data they contribute. In our environment, we offer visualization and exploration applications for advanced analytics, and we connect experts in support of accelerating discovery. Here you see a more detailed explanation of what the BRAINCommons has to offer in our visual framework. Our governance and oversight component supports our data contributions, user authentication and onboarding, data and compute access requests and approvals, as well as our data management and permissioning. Then you have our data ingestion, storage, and access component, supporting our data inventory, upload, curation, and harmonization process, where the data is then stored for access in the appropriate zone. Zone 1 is public, Zone 2 is controlled, and Zone 3 is restricted. Then, of course, you have our collaboration, community, and computation components that support our end users' ability to explore, analyze, and share. Underpinning all of this is, of course, our security and compliance component, supporting NIST CSF standards and complying with GDPR regulations. In the next slides, we'll dive deeper into each component of this framework. Leveraging interoperability approaches that harmonize content and format of data from multiple studies or projects, we increase the value, enabling the creation of new cohorts for additional analysis and discovering of meaningful insights. The fact that all BRAINCommons data is curated and harmonized to the BRAINCommons common data model is one of the major value propositions of the platform. And the next slide will provide a bit more information on the advantages of modeling the data. If you would like more information on the actual model or our plans for the future, please be sure to reach out to me using the contact information provided at the end of the presentation. The BRAINCommons is ingesting data from multiple studies and harmonizing to fit a structure that can be easily queried, also known as semantic interoperability. Without a data model, the data would be collected into one giant table without relationships or links between the different types of data. You can think of it as laundry in a basket that hasn't been sorted versus laundry that has been sorted for washing. It makes your whole process so much easier. The advantages include things like our ability to leverage major models and standards that allow us to participate in an open source community aiming to standardize health data more broadly. We ensure data consistency and quality within and across studies. We enhance sharing of our BRAINCommons data using models that follow the FAIR principles for findable, accessible, interoperable, and reusable data. In the end, we're contributing to hypothesis generation, data exploration, replication, validation, and systems modeling for brain health, all with our tactics around semantic interoperability. Now we'll direct our focus to the zones of the BRAINCommons. This is where the data is stored for access once it has been uploaded, curated, and harmonized to the BRAINCommons data model. Zone 1 is called the public zone, Zone 2, controlled, and Zone 3, restricted. There are 3 zones where data can be stored for access in the BRAINCommons. Zone 1 is open to the public, which means all users. It only requires an active email, and a user can register an account in the BRAINCommons after accepting our terms of use and privacy notice. Zone 2 is our controlled zone. This zone is for qualified researchers. Qualified researchers are researchers holding a research position at an institution of higher education, a research organization, a nonprofit organization, or a government agency. In this zone, once approved as a qualified researcher, a user has access to all data in this zone. Our approval process requires completion of a 45-minute training module, signing of a data use agreement, and approval from our data access committee. This entire process takes about 2 weeks to complete, and these users are also eligible for a workspace. Zone 3 is our restricted zone. This zone allows contributors of data to select who can access and use the data. This is provided on a project-by-project or dataset-by-dataset basis and has the same requirements as Zone 2, also making these users eligible for a workspace. Now we'll shift our focus to the collaboration community and computation component of the BRAINCommons. This allows users to explore the projects or cohorts, our data model, and the different data types available, as well as the tools we offer to support analysis. Users can leverage BRAINCommons workspaces with notebooks and can upload custom pipelines or tools of their own. Lastly, we offer users the ability to share their notebooks and results with their teams or the broader community. The BRAINCommons is enabling exploration, analysis, and multidisciplinary collaboration for insight and discovery across data sets and diseases in support of brain health. We offer secure, custom workspaces with pre-configured tools such as Jupyter Notebooks with RStudio and Python coding capabilities, all of this allowing multidisciplinary users the ability to have sophisticated visualizations and conduct analytics that illuminate the value of data, facilitating computational modeling, simulation, and machine learning, supporting a systems brain model, and sparking global collaboration and enabling innovation. Of note, CVB didn't just spearhead the development of the BRAINCommons. It is also a primary customer, sharing several deep phenotyping clinical studies. In the next few slides, I'll share a use case with you to illustrate the potential for pan-cohort analysis using the BRAINCommons. A normative neuroimaging library, or NNL, as I will refer to it moving forward in these slides, is an essential aspect to the development of effective clinical imaging tools for diagnosing and managing patients with brain disorders. Quantitative MRI holds enormous promise in advancing diagnosis, prognosis, and treatment planning, but requires an understanding of the characteristics of quote-unquote normal population, which does not readily exist for any forms of imaging such as diffusion, perfusion imaging, volumetric analysis, and functional MRI. So what is normal? The lack of normative data remains one of the largest obstacles for the clinical implementation of many of these advanced modalities. We cannot use quantitative data without a reference range, as is necessary for other lab and cognitive tests. To overcome this obstacle, CVB is funding the development of an imaging reference library, NNL, to advance identification of affected individuals. The PIs of the project are listed here: Dr. Elizabeth Wild, Dr. James Stone, and Dr. Stephen McCauley. Data collection has been happening for more than 5 years with a goal of 3,000 participants. To date, the study has collected data from more than 1,800 individuals. In the next slides, I'll provide a bit more information about the clinical endpoints and neuroimaging modalities and demographics captured. Here's a list of the variables that were identified as having the ability to influence the neuroimaging. These include age, gender, race and ethnicity, handedness, socioeconomic status, IQ or cognition, psychological health or psychiatric disorders to include substance use, and medical history. I'll give you a bit more details on the medical history components a little bit later in the slides. Here I've listed the neuroimaging modalities used on the left. These include T1 and T2 weighted, susceptibility weighted imaging, FLAIR, resting state, fMRI, and arterial spin labeling. On the right, you can see the standardized imaging protocol, starting with human phantom. I've listed here the 3 phantoms, and then that it's run monthly. If you have particular questions on any of these items, I've got my contact information at the end of the slides. Please be sure to reach out. As I mentioned before, here are some details on the information collected related to medical history. We've included mother's pregnancy history, developmental, exposure to concussion and TBI, obesity, cancer, lead exposure, history of seizure disorder or neurodegenerative disorders, cardiac disease, renal liver disease, headache, general anesthesia, medications, diabetes, hypertension, sleep apnea, asthma, substance misuse or dependence, chronic pain, and psychiatric disorders. And here, additional details on the information collected related to cognitive measures and behavioral assessments, performance validity testing, general intelligence, memory, attention, executive functioning, processing speed, pain, insomnia and sleep, PTSD, mood, including depression and anxiety, post-concussive syndrome, and alcohol and substance use. Lastly, the NNL study group decided to add new data types at the end of March 2021. These included blood draw for clinical labs and potential candidate biomarkers, ADHD self-report rating scale, a COVID questionnaire, and gender and related hormone therapy. The BRAINCommons is creating an opportunity for cross-cohort analysis using NNL as a reference database to serve as a key resource for the development of effective clinical imaging tools for diagnosing brain disorders. This is a bit of a busy slide, but I think it does a good job of summarizing basically the whole premise here. You can see that NNL data can be combined with other data sets that are accessible via the BRAINCommons. These would be listed data set 2, 3, or 4. You can also obviously think of them as cohorts. Because of our ability to curate and harmonize the data across all of these cohorts, the data is then ripe for analysis and discovery. At the end of this whole process, you are then generating results that can be shared with those within your team or across the larger BRAINCommons community. The BRAINCommons is the environment providing researchers and data scientists the research-ready data needed to execute end-to-end workflows to answer questions that can reduce the burden of brain disease and promote brain health. In summary, you can see the end-to-end workflows here in this diagram. Users can access the platform via highly secure infrastructure using cybersecurity best practices, get a bird's eye view of the distribution of data by zones, disease, etc. across projects or cohorts. They can view a project gallery to explore the metadata about the cohorts that have been contributed to the platform. They can browse the Data Explorer to create and save a custom cohort from combining data from several cohorts using faceted search features with an easy-to-use dashboard. They can export and visualize harmonized data on secure workspaces offering Jupyter notebooks and RStudio. They can analyze data using example scripts to query data via GraphQL and import custom pipelines to run in-platform analysis. And then finally, share results via workspaces using team-based approaches to then export results for publication. I hope you enjoyed this introduction to the BRAINCommons and the example of how an existing data set such as NNL can use the types of capabilities offered by the BRAINCommons in support of pan-cohort analysis. With the BRAINCommons, we are able to accelerate pan-cohort analysis and contribute to the field of brain health. Again, my name is Maryan Zirkle. I've listed my contact information here at Cohen Veterans Bioscience. Please reach out to me with any questions or requests for additional information. Thank you.